Hi, my name is Dr. Abigail Johnson, and I'm a registered dietitian and postdoctoral fellow. I work with Dr. Dan Knights to study diet and the microbiome. You might have heard of the microbiome, but you might not fully understand what it is. So this picture that you can see on the screen right now is actually a representative image of what we try to talk about when we talk about the microbiome. This is a false-colored electron microscopy graph of the microbiome, and it clearly shows the diversity in the community of bugs that live within your gut microbiome. And what's also visible in this picture is this stick-like object that runs diagonally through the image. And this looks like it might be fibrous material from some sort of plant. And I really like this picture because it really gets across the point that the microbes that live within us have to coexist with the diet and with the food that we feed ourselves. So the bugs that live within us use the food that we eat, and in return, what we're now learning is that they also send out signals that interact with our body's physiology in ways that we never imagined that they would. So my objectives for this lecture today is to go over what the microbiome is, explain how we know which bacteria make up the microbiome, and I'm going to do this through discussing classification broadly, touching briefly on 16S sequencing, also talking about shotgun sequencing or metagenomic sequencing. And then I'm going to talk a little bit about what we can learn from the microbiome, what we don't know about the microbiome, and which challenges exist for research and technology in relation to diet and the microbiome. So what is the microbiome? One of the definitions that I like is that it's the community of microorganisms that occupy a specific habitat. So in nutrition, we typically focus on the human gut microbiome. And so this image here is a picture of your gut, and there are microbes that live within your stomach, your duodenum, your jejunum, your ileum, but the majority of the microbes that live within us live within the colon or the large intestine. In fact, there are more gut bacteria or bacterial cells within your gut than there are cells within your whole body. So when we think about the microbes that live within us, they are potentially very impactful. So how do we know what makes up the microbiome? Obviously, we cannot see these bugs with our eyes. And for a very long time, the microbiome was viewed as this sort of black box. We knew that we put food in, and we knew that there were communities living within us, but we didn't understand which bacteria were living within us. And partly this was because for a long time, we couldn't grow the bacteria in a petri dish. So thanks to the pioneering work of a microbiologist named Norm Pace in the 1980s, we realized that we could sequence the bacterial DNA from microbiome samples or human microbiome samples like poop, and we can then use that sequence information to identify all the bugs that live within the human gut without ever having to culture anything at all. So before we can talk more about this, I'm going to go through a quick refresher of what classification is, or phylogenetic classification. And don't worry about the jelly beans, we'll get back to them in a minute. So when you think about trying to classify an animal, we know that they can be classified by kingdom, phylum, class, order, family, genus, and species. So this is an example with, say, the platypus. You can see the classifications for this platypus down to its species name. Oh, Anatinus. So we can also do this with bacteria. This is an example of the classification scheme for E. coli. And you might be familiar with E. coli because we talk about it a fair amount in food safety or when people get sick from E. coli. So in the case of E. coli, we can classify it as bacteria, proteobacteria, gamma proteobacteria, enterobacteriales, enterobacteriaceae, Escherichia, and then E. coli, which is the name we usually use to talk about the species. There's actually another level, though. We can classify E. coli to the strain, which is when we start to talk about which types of E. coli are pathogenic to humans. So remember these jelly beans? What do the jelly beans have to do with the microbiome? Well, it's not always easy to know which flavor of jelly bean you're going to get when you reach into a container. Even if you can see the color, you can't always tell what flavor the bean will be until you taste it. This is more of a problem if you know someone who recently visited Harry Potter World. because they might have added some every flavor beans to your jelly bean jar. So we face similar issues when we want to know what species of bacteria live inside our gastrointestinal tracts, and we've developed methods to work out what they are from their genetics. At a macroscopic level, this shares similarities with working out which flavors are in our jelly bean jar. So I'm going to go through a little thought experiment with you here. Luckily, when it comes to jelly beans, we have a key, and we can match them visually to the flavors. But we don't always do this perfectly. So if I wanted to group my jelly beans to describe the composition of my container, I might choose to classify them broadly by color first. And this is similar to what we do with microbiome data. So how does classification work? We can look visually at our beans and put them into a color category. Some will be harder to categorize than others, but we'll have to assign them a category. So Tutti Frutti, which has similarities to many colors because it's multicolored, we'll have to pick a color, and in this case I might pick pink. Or strawberry cheesecake and strawberry daiquiri obviously have very similar colors, so they're going to be classified the same. So this is what the classification might look like once we're done putting them into color categories. You can see we've lost some detail, but we now have a way to group our beans easily. So we can look at the distribution of the colors and count up the number of times that we find each color in our sample, and then divide by the total of number of times that we assigned a color. In this case, we have 8 categories, 50 total assignments. So we can also view this data a separate way. We can look at this with a stacked bar plot. This captures the percent of each color category in our sample. So in this case, we've got 8% white, 10% pink, 10% black, 12% blue, 14% green, 14% orange, 16% yellow, and 16% red. But we don't have all the detail, but it does still tell us about the general makeup of a jelly bean jar. So if I were to pass my jelly bean jar around a classroom and ask you all to take out some jelly beans, you'd all have different preferences. And if I repeated this with 2 different classes and then resampled my jelly bean jar, I would get 2 different representations of the preferences of the class that ate the jelly beans. And they might look like this, 2 very different sets of stacked bar charts relative to what we started with. So when I think about real microbiome samples, we get the same sorts of data, but this time we use classifications like phylum or genus or species, like we talked about earlier. So in this case, this is data from the Human Microbiome Project, which you've been assigned to read about as part of your in-class assignments. Here you can see that bars, like those that I showed you for the jelly bean samples, are visible with big differences between the sites. So on the far left, you have samples from somebody's nose, and on the far right, you have samples from stool and fornix. As you can see, there are vast differences between the microbial samples that live in your nose and those that live within your gut. So how do we get this microbiome data to begin with? Well, first we need to extract DNA from a sample. Then, once we've extracted the DNA, we need to amplify that DNA. And then we can sequence our samples with a machine like the one shown here. And what we get after we've sequenced samples are computer files that look like this. Lots and lots and lots of A, T, Gs, and Cs representing the bases that make up the DNA of the microbes in the sample. And once we have those DNA sequences, we can use computer software like one called QIIME, and then we can align those sequences to a database. And then we can do further data analysis in programs like R or Python. But there is a choice that we need to make when we amplify the DNA. We need to choose whether or not we're going to use 16S sequencing or if we're going to do shotgun or metagenomic sequencing. So 16S sequencing or barcoding uses a region of the genome that every bug has, which is called the 16S ribosomal subunit. Essentially what you do is you make lots of copies of this single gene, which you can see here on the picture on your screen outlined in the box in red, and you line up all of these copies of this gene with a computer and compare them to known 16S sequences from every bug that we know that lives within your gut. And this allows us to make comparisons like apples to apples because we only have one piece of the genome that we need to align. The pros of using this method is that it's cheap, but the con is that we usually need more sensitive techniques if we want to identify strains of bacteria. So if we go back to the jelly bean example, if I want to correctly assign the flavor, I need a more sensitive method than the color method that we discussed previously. In this case, I can taste the jelly beans, and I've increased the sensitivity to be able to distinguish the flavors by using my tongue. But it will take a little longer. So this is kind of analogous to whole genome sequencing, or shotgun sequencing, or metagenomic sequencing. Essentially what we do is we take the DNA from our sample, we break it up into lots of tiny little pieces. This is where it gets the name shotgun. And then we sequence all the tiny little pieces and look for the overlap between those pieces, which we can then align to all the parts of known reference genomes. But this is a much more computationally intensive task. So, with this kind of shotgun sequencing, for a very long time, the con was that it was too computationally costly. You could ask a computer to complete the task of alignment, and it would take days or weeks or months to be able to finish the alignment. So you need very smart computer programs to do this. We also need databases of full genomes. Luckily, we have this now for most of the bugs that live within the human gut. There are about 1,000 species. So now that we can begin to do this, we have some computer programs that are able to do this efficiently. So the cost is coming down, and the big benefit of this type of sequencing is that it allows for higher specificity. So we are able to identify strains of microbes, and we're also able to learn about bacterial function because we're not limited at looking at just the one gene that we've sequenced, like 16S ribosomal subunit. So what can we do with this data? Well, we can make comparisons between different health and disease states, which is what a lot of people are very interested in doing right now. We can also determine if the community that lives within our guts responds to changes and differences in our environment, like diet, where we live, antibiotics that we take, probiotics, which are bugs that you would consume, and then they would take up residence in your gut, or prebiotics, which are things like fibers that you would consume that then feed good bugs differentially. Ultimately, our goals are to find mechanisms for how microbes interact with our biological systems. So how does diet impact the microbiome? And I don't expect you to look very closely at these pictures, but these are example pictures from a very nice review that's available in the literature if you want to read more. And there will be a link in one of your class worksheets for this. So from this review, we know that there are interactions between the diet and the microbiome, and that the majority of our understanding right now focuses on fiber and the generation of things called short-chain fatty acids from the breakdown of that fiber to specific molecules like butyrate and acetate. And these small molecules are the end product of that microbial fermentation of fibers. And they can act as fuel for the cells that line your gut, but they can also contribute to various signaling pathways and have wide-reaching effects on peripheral tissues within your body beyond the gut. And we also know that diet very quickly changes the microbiome. So you don't need to worry about the specifics of this picture, but what this shows is the green portion is an intervention diet. And what you can see on the second graph here is that when the diet is changed to something that the people were not used to eating, the microbiome very quickly responds and changes within a matter of days. So we know that big shifts in diet can cause big shifts in your microbiome composition. So the question then becomes, can we predict how you will respond to food if we know your microbiome composition? And there's a recent study that looks into this, and it's by Zivi et al. And the title of the study is called Personalized Nutrition by Prediction of Glycemic Responses. And it's published in the paper in the journal Cell. So this made a very big splash when it hit the scientific community, because these authors showed that if we knew a person's microbiome and we knew some information about their usual blood glucose levels, then we could predict with some degree of accuracy how they would respond to a particular carbohydrate-containing food. So for example, one person might eat a banana and their blood glucose might spike, but for another person they might eat the same banana and their blood glucose would not spike. So what these authors predict is that they can look at your microbiome and then predict whether or not you will respond positively to a banana or a cookie, for example. And I do encourage you to read this paper if you're interested in going deeper into this subject matter. So now the caveat to all of this prediction and understanding the microbiome and the diet is that we don't know what healthy looks like. So if you remember this picture that I showed you previously, the area called stool on this graph shows this massive variation. On the left side of that panel, you can see there's a huge amount of blue in some of these individuals. So every bar in this graph is one individual. And these are all very healthy people. So the left side of this graph, those people are almost entirely populated by blue, which is Bacteroides. On the right side of this graph, those people are almost entirely populated by a different type of bug from Firmicutes. So we don't actually know what healthy microbiomes are because there's this huge spectrum of what healthy looks like. Another way to sort of hammer this point home is some research I've been recently doing where we've collected microbiome composition longitudinally in individuals over time. We've also collected their dietary information. So this picture shows you on the left the food composition of the diets that the people ate during the study, and on the right, the microbiome composition over the course of a study. And what you can see is that microbiome composition changes a little bit every day over time, but not drastically, whereas the food composition changes all the time. So we're trying to figure out ways that we can tease apart this very complex series of interactions because we still don't understand how it all works. So how is this kind of science being sold to consumers? You might have seen some of these books if you go on Amazon. There is a book for feeding your microbiome at every level and price range. And many people are trying to tell you how to eat to feed your microbiome. But the reality is our research isn't really backing up what these people are saying. You can also find information about the microbiome diet if you look online. Again, these are kinds of diets that haven't really been backed up by science yet. So our ability to understand and support these claims with data is still very limited. So we can't really modulate or isolate particular foods and the resulting changes in your microbiome yet. And we really don't know what makes for a healthy microbiome when it comes to diet. So if you remember this paper that I already mentioned, these authors, after publishing this paper, started a company. And this company is called Day 2. So if you want, you can pay this company to sequence your gut microbiome, and you can also send them a sample of your stool and have a blood test done at your clinician's office. And then they'll take that information and they'll sell you an app. And this app will tell you what to eat based on your microbiome and your blood glucose. So the things I want you to think about when you're doing your assignment is to question whether it's possible to leverage what we know about the microbiome and diet to translate microbiome technologies into a service. I want you to think about if it might be too early to do this with the current data that we have available, and if so, why or why not. Also, I want you to think a little bit about if you feel we know enough information to be charging money for these types of services. And then if there's any chance of harm. I also want you to consider if our recommendations that we make for a healthy microbiome diet, despite not necessarily being based in science, are generally in line with what we know to be a healthy diet. Does it really matter if we don't know what the impact of on our microbiome might be, if it results in positive behavior change? And then finally, I want you to think about what the role should be for dietitians in this field and where we might fit in this changing landscape. So if you want to learn more, you can visit the Knights Lab website, knightslab.org, or you can feel free to contact me by email at cole0463@umn.edu, and I'd be happy to talk to you further.